Good day, Flame Photometry Module 8. Today we are going to learn about principle and instrumentation of flame photometry. Flame photometry is also called as flame emission spectroscopy because neutral atoms are involved in the emission of radiation when introduced into the flame. So it is also called as flame emission spectroscopy. Let us see the principle involved in flame emission spectroscopy. When you take a solution of metallic salt and it is sprayed onto a flame, a fine droplets are formed. Due to the thermal energy of the flame, the solvent in the droplets evaporate, leaving behind fine residue. You can observe the diagram. This residue is converted to neutral atoms. These neutral atoms are converted to excited state atoms by the thermal energy of the flame. As the excited state is not stable, these excited atoms return to ground state with the emission of radiation of specific wavelength. Here you can observe the diagram. This is the sample solution which is sprayed onto the flame and produces a fine residue, leaving the solvent in the droplets. And this fine residue is converted to the neutral atoms, you can observe. As these neutral atoms, with the help of thermal energy, they undergo excitation from ground state to excited state. As this state is unstable, again these excited atoms returns to ground state with the emission of radiation of specific wavelength. The wavelength of radiation emitted is characteristic of the element and is used to identify the element. So it is a qualitative analysis. And also the intensity of the radiation emitted depends upon the concentration of the element analyzed. This is quantitative analysis. Hence, flame emission spectroscopy can be used for both qualitative and quantitative analysis. Let us see the instrumentation. The basic components for flame photometer are burner. It contains fuel and oxidant as a source, atomizer, monochromators or filters, detector, readout device. Let us study one by one. First one, burner. There are different types of burners available which are used to spray the sample solution into fine droplets mixed with the fuel and oxidant, so that a homogeneous flame of stable intensity is obtained here. The most common using burners are total consumption burner and laminar flow burner. You can observe the diagram here, the total consumption burner. The construction of burner is very simple here. In this type of burner, the sample solution is aspirated through the capillary. You can observe the diagram. This is the sample solution which is aspirated through the capillary by the high pressure, and the fuel and oxidant is also present here. And this will be burnt at the tip of the burner. The advantage with the total consumption burner is very simple, easy to operate and inexpensive. But the disadvantage with the total consumption burner is it doesn't produce a uniform and homogeneous flame, since the droplets may vary size and this leads to fluctuations in the flame intensity. Next, coming to the laminar flow burner. This burner is the most widely using burner because of its uniformity in the flame intensity. In this type of burner, the sample solution, fuel and oxidant are mixed before they reach the burner tip. You can see the diagram. This is the sample solution, oxidant and fuel, and they enter into the burner, and before reaching the flame, this is the flame, they will mix in the burner, and therefore only few droplets of uniform size reaches the flame and remaining are drained out through the outlet at the bottom. And this is the most commonly using burner because of its advantage, uniformity in flame intensity. And here different types of fuel and oxidants are also used in the burner, like propane, hydrogen, acetylene. Next, atomizer. Atomizer is a device that produces a fine spray from a liquid. Also, it produces a homogeneous spray. So, it is used here. Next, coming to the filters or monochromators. In flame photometry, the wavelength as well as intensity of the radiation emitted by the element is monitored. Hence, filters or monochromators are used. The function of filter or monochromator is it converts the polychromatic radiation into monochromatic radiation. In the simple flame photometer, filter wheel will be present for calcium, lithium, sodium or potassium. When a particular element has to be analyzed by using flame photometer, the specific filter can be selected and we can estimate the sample. In this way, we can use these filters and monochromators. For detailed description, you can go through the link. Detectors. The detector function is the radiation emitted by the elements is detected in the readout device. And here, the radiation which is emitted by the elements is mostly in the visible region. Hence, conventional detectors like photovoltaic cell or phototubes can be used. And most commonly in the advanced instruments like flame spectrophotometer, photomultiplier tube is used as a detector. And you can go through the link for the 3 types of detectors, that is photovoltaic cell, phototubes and photomultiplier tubes. Next, coming to the readout device. The signal from the detector is shown as a response in the digital readout device. The readings are displayed in the arbitrary scale, that is percentage of flame intensity. So this is the instrumentation of flame photometer or flame spectrophotometry. And in the instrumentation, we have studied about the burners, atomizer, filter or monochromator, detector and readout device. Let us see the instruments used in the flame photometry. So this is the instrument which is a single beam. And you can see the schematic diagram here. The components already we have discussed, that is the burner, flame, fuel and oxidant, filter or monochromator, detector and readout device. You can see in the diagram. And here the flame photometer contains the burner where the solution to be analyzed. So this is the burner where the solution to be analyzed. And it is sprayed into fine droplets or after mixing with the fuel and oxidant. And a compressor compresses the air or oxygen and fuel at a pressure. This high pressure is required to spray the sample solution to fine droplets. When the sprayed droplets are ignited, the solvent is evaporated by the thermal energy of the flame, leaving behind fine residue. This fine residue is converted to neutral atoms. The neutral atoms are excited by the thermal energy of the flame to excited atoms. These excited atoms returns to ground state. Finally, the wavelength and intensity can be measured by using the filter and photometric detector. This is the filter and this is the detector. And finally, the signal from the detector is shown as a response in the readout device. This is the flame photometer. Next, coming to the flame spectrophotometry. And here in the flame spectrophotometry, 2 types will be there, single beam and double beam spectrophotometry are available. And you can observe this is the double beam spectrophotometry. In this spectrophotometry, already we have studied the components regarding the burner, fuel and oxidant, filters and monochromators and detectors. In this spectrophotometry, the radiation from the flame passes through 2 different filters or monochromators. You can see here. The radiation that is coming from the flame is split by the beam splitter and it falls one for the reference and one for the sample. The double beam configuration is used to monitor a sample element and an internal standard. So in this way, we can analyze the sample by using double beam flame spectrophotometer. And it is used for the quantitative analysis. So these are the instruments available with the flame photometry.